Hi everyone, this is Miss Chung. This is the first recorded lecture, and the point of this is to make the textbook reading easier. So I'm going to give a brief overview of 5.1 to 5.6. I encourage you to take notes, and so hopefully by listening to this PowerPoint and taking notes on this, you could just skim the textbook and this should pretty much cover a lot of the big ideas. I still encourage you to read the textbook, but this should make it easier. Okay, so we'll start. All right, the two big ideas that you need to know about the membrane is that it's described by the fluid mosaic model and that it's selectively permeable. Those are the two big ideas about the membrane in terms of its structure, and we will describe them in just a bit. Okay, so for the fluid mosaic model, what this means is that the membrane is made of a phospholipid bilayer. So those phospholipids, there's two layers of them that are right next to each other. But not only that, there's proteins that are embedded inside the membrane. And so because there's so many different types of proteins floating around, it's called mosaic. That's the mosaic part of the name fluid mosaic model. But it's also fluid because these proteins can kind of move around. They're not like stuck in one place. They're kind of shifting around, and the membrane itself is very, it's almost like liquidy. It can kind of float and it changes its shape and it's very like malleable. So that's why it's called the fluid mosaic model. This right here is just a picture. You can see the phospholipid bilayer. Yeah, there's different types of proteins that are embedded. And notice that these proteins that go through the membrane, they're called transmembrane proteins. They have both hydrophobic and hydrophilic parts. And it should make sense to you from the picture which parts are where. Okay. Let's see. So the reason why
Camtasia will
the
export it.
membrane is so fluid or flexible is because those phospholipids, they have they're unsaturated. So those fatty acid tails kind of have kinks inside of them due to the double bonds. And so because of that, they can't pack so closely together. And so because they have all these kinks and everything, they're kind of like a little bit, there's space between each of the phospholipids. And so they kind of move around more fluidly. And the other reason why it's fluid is because there's actually bits of cholesterol, which is a different type of lipid, and they tend to be inserted inside the membrane, and so they kind of they're also something that helps keep those phospholipids separated from each other and helps them to move around more fluidly. Right here is a picture of those kinks you can see. You can't see the double bond directly in the picture, but those fatty acid tails are kind of move off to the side like that because there's a double bond there. So there's kind of space. Okay, in terms of the mosaic part, the different types of proteins you can find in the membrane. There's a lot of them. On this PowerPoint slide, you can see the names of the different types of proteins you can find in a membrane. And these are different ones that you're going to just kind of have to know. And I would encourage you to read through these and write these down in your notes. This is just a picture to describe all those different types of proteins. You can see all the way to the left-hand side. This right here is an attachment protein. And we just learned in the last chapter that this could also be called an integrin. And you know that this is an integrin because it's attached to a glycoprotein here, which is attached to the ECM or the extracellular matrix. This one's a receptor protein. Usually like ions or different things will attach to it, and it gives a signal to the cell that it needs to start some kind of reaction or some kind of chemical pathway. So as soon as the receptor binds to this protein right here, that it kind of leads to some like chemical pathways afterwards. This right here is just a channel protein and allows certain types of molecules to pass through. This is an active transport channel protein, which means it's the same idea. It allows certain molecules to go through, but it requires energy. Here's a junction protein right here, and it just binds with other junction proteins of another molecule, and it allows those two cells to kind of bond together. And then right here is a glycoprotein, and this sugar tag on the top of the protein, the glyco part of it, can kind of be read by other cells, and it helps the cells get to kind of figure out what that cell is. It's kind of like its name tag or something. Okay, the second big idea aside from fluid mosaic model is that membranes are selectively permeable. That means that these membranes only allow certain things to pass through easily and other things it kind of keeps out. And so there are a couple things that affect its permeability or its ability to accept certain things. Number one, it's very much affected by the polarity of a substance. So because those membranes, the majority of that, the phospholipid is that fatty acid tail, right? Nonpolar molecules can pass through that membrane very easily, small nonpolar molecules. Whereas polar molecules, anything that's charged, it can't just pass through that membrane because it can't pass through the fatty acid tail part of the membrane easily without any kind of help. So nonpolar passes through easily. Also small molecules. So if the size of the molecule is really large, it just can't slip through that membrane very easily. Whereas if it's very, very small, it can. And hydrophobicity is very similar to the polarity of the molecule. So at the end of the day, small nonpolar molecules can pass through easily without any sort of help. Whereas if it's larger or if it's polar, both of those things, it can't pass through easily and it needs to have some kind of help to pass through the membrane. From section 5.2, all you really need to know is that phospholipids form spontaneous membranes, and so the theory is that this kind of helped early life start to form. And membranes were very important to life because it helped regulate the chemical exchange between the internal environment of the organism and the outside environment. Okay, so we're going to be talking about in this section 3 major ways for things to get inside of the cell through the membrane. It can either do passive transport, which means it requires no energy and it can just get through. It could either be active transport, which means that it has to require energy to get through the membrane. Or it can kind of go through cytosis, endo or exo, which means that the membrane kind of swallows like things from the outside and forms a vesicle. A vacuole around it, and it kind of gets transported inside the cell or outside. Okay, so before I start talking about the different types of transport through the cell, I'm going to describe a little bit about the natural process of diffusion. Diffusion is simply just the process where the particles spread evenly throughout a space. And so diffusion happens naturally, and particles tend to move from where they're more concentrated to an area that they're less concentrated. They want to be even throughout, right? And that process of it going from more concentrated to less concentrated is called moving down the concentration gradient. It likes to move down the concentration gradient. And eventually, it will reach a place where the number of particles are the same on both sides. That's called equilibrium, but it's dynamic equilibrium because even though the number of particles are the same on both sides, they're constantly kind of switching places with each other, but overall, the numbers will be the same. So that is called diffusion. Passive transport is the name that we give the type of diffusion which does not require energy. So passive transport is pretty much the same thing as diffusion. It doesn't require any energy, and the molecules will always move down the concentration gradient from more to less. Here's just a picture. If you start off with a bunch of molecules on one side of the membrane, they'll start to move over to the other side, right? All of these molecules are moving around with kinetic energy, and eventually they'll move around to the other side. And here you'll eventually reach a state of equilibrium where the number of molecules will be the same on both sides. But remember, they're still moving, so some of them might switch places. This one might go on this side, this molecule might go on that side, and they keep on going back and forth. But overall, the numbers will stay the same. Just take note that you might have 2 different types of substances, right? And they diffuse independently of each other, which means that eventually all these purple guys want to be equal on both sides, and the green guys right here want to be equal on both sides. So at the end of the day, you will get the same number of green on both sides and the same number of purple on both sides, roughly. Okay, osmosis is the specific name we give passive diffusion for water. And so just like we were talking about, water tends to move where it's more concentrated to less concentrated. And it's a certain type of diffusion. Let's see, this right here is just a picture of osmosis. So this right here, this gray dotted line, is a selectively permeable membrane, and it doesn't allow these purple solutes to move. So if these purple things can't move, the only thing that can move is water. And water will distribute itself so that it goes to a place where the concentration on both sides of the solution will be the same. So here, do you notice because there's more molecules here, less molecules here, the water will actually move from where there's less molecules to where there's more molecules. And now do you notice that the concentration, and remember concentration means the amount of solutes per amount of solution, is about the same here and it is about here. So it diluted this side and it made this side more concentrated by moving the water over. Here's a picture. And your textbook reading describes this on a molecular level by saying that if you think about all these little molecules that are surrounded by water, that frees up less of the water here. And so water is technically, there's more like free water molecules in the area that has less solutes. And so the free water molecules will move over to the side where there's less free water molecules. Because remember, if there's more solutes here, they're going to be all the water molecules will surround them. And so there'll be less free water molecules. Okay. So water balance is really, really important for cells. And tonicity right here is the word that we use to describe whether or not solutions gain or lose water. And you can have 3 different states of tonicity. You could have isotonic, which means the solution has the same concentration of solutes on both sides. You could have hypertonic, which means that the concentration of solute is much higher than what's inside the cell. And then you can have hypotonic, which means the concentration of solutes is much lower than the concentration inside the cell. So osmoregulation right here is the word that we use to describe maintaining the water balance. And so overall, cells want to make sure that they have the right amount of water. And animal cells and plant cells want different amounts of water, and you'll see that in just a second. This is a really important picture, and I would take some time to really look at and understand this. Isotonic solution is when the same amount of water is entering as is leaving because the solute concentration is the same on both sides. This for an animal cell is ideal. This is what the animal cell wants. Actually, for the plant cell, when you have the same amount of water entering and leaving, it's called a flaccid state, and the cell does not like that. It tends to be kind of limp when things are like that. Hypotonic solution, that's when there's less solutes on the outside than there is on the inside. And so remember, if there's less solutes on the outside, that means the water is going to want to rush inside the cell to make the concentration levels about equal. Now, when water rushes inside the cell, for an animal cell, that's really bad. It gets too full, and then the cell actually bursts, and it's called lysed. Whereas actually for the plant cell, because it has that rigid plant cell wall, when it's in a hypotonic solution where the water is rushing inside the cell, it becomes nice and firm, and the state is called turgid. And that's actually the ideal state for the plant cell. So the plant cell wants to be turgid, whereas the animal cell wants to be normal in an isotonic solution. Plant cell wants to be in a hypotonic solution. And the last state, which is bad for both animal and plant cells, is called hypertonic. That's when the outside of the cell is very, very concentrated with solutes. Like imagine if you put your cell in like a bunch of salt water. That would be like a hypertonic solution. And so when it's surrounded by something that's so concentrated, the water will leave the cell and go into the environment. And when that happens, it actually really shrivels up the animal cell and it gets all like very thin and wilted, and this actually kills the cell. In the same way for the plant cell, water will also leave from inside to outside to try to make the concentration of the outside cell a little bit more dilute. But when it does that, you actually have something called plasmolysis, or it gets plasmolysed. That's when the cell membrane, it's trying to collapse in the same way that the animal cell does when all the water leaves. But when it does that, it detaches from the cell wall. And so as it shrivels up and detaches from the cell wall, the plant cell dies. Okay, last section. We're still talking about passive transport. We talked about how osmosis is a type of passive transport, which just involves water. Another type of passive transport, which means it requires no energy, is something called facilitated diffusion. So this is a type of diffusion that is the same thing. It requires no energy. The molecules still move down their concentration gradient, so from more to less. But the only difference here is that it requires a special type of transport protein to help it move across the membrane. And so remember, the reasons why it might need a transport protein is that it's either polar or it might be very large. So either of those things might requires a protein to help it to move through. These transport proteins, they're another word for them is channel protein. And the specific type of transport protein for water is called aquaporins. And remember, water is a very polar molecule. Even though it's small, it needs a transport protein called the aquaporin to go through the membrane. This is just more information on transport proteins. And here's a picture right here. You can see that it kind of goes through the membrane. And so you can imagine out here is the hydrophobic parts. But inside, coated in here, might be hydrophilic if, for example, the molecule it needs is a polar molecule. And so the polar molecule can move through the inside of it because it's hydrophilic and go from where it's more concentrated down to where it's less concentrated. Right. All right, that's the end of the section 5.1 to 5.6. Thanks so much for watching, and I hope that this makes things easier to read the textbook.